Alrighty, so let's have a brief discussion on compartmentation, like cells and tissues. So this is going to be chapter 3 of physiology, right? So let's talk about that. So we're going to talk about functional compartments of the body, right? Biological membranes, intracellular compartments, tissues, remodeling, and some organs, right? Just general. So the functional compartments of the body, right? So we have 3 major cavities: cranial cavity, the thoracic and the abdominopelvic cavity. And so these fluid-filled compartments, we're going to have the circulatory system, we're going to have the eyes, right, cerebrospinal fluid, and the pleural or the pericardial sacs, right? These are going to be fluid-filled compartments within the body, right? So the lumens of some organs, they're going to be outside the body, right? So for example, the GI tract, right? So our hollow organs, we're going to have the heart, the lungs, blood vessels, and intestines, right? These are going to be hollow organs. And the lumen, right? The lumen is just going to be the interior of any hollow organ, right? They may be wholly or partially filled with air or fluid. But like I mentioned first, some organs, they're going to be really an extension of the external environment, such as the GI tract, how we have that opening, right? Excuse me. From the mouth, so like cheek to cheek, right? It's really an extension of the external environment, because what comes from the external environment gets into our body like that through our GI tract, right? And it exits through our GI tract as well, okay? For example, okay? And so functionally, the body has 3 fluid compartments, right? We have intracellular fluid. This is going to be the fluid inside the cell, okay? And extracellular fluid. This is going to be a fluid outside the cell. And so there's going to be 2 compartments of extracellular fluid. There's going to be interstitial fluid, right? This is interstitial fluid, right? The fluid that surrounds most cells. And then we have plasma, right? That's going to be the extracellular fluid that lies within your blood vessels, okay? So intracellular fluid is going to be the fluid within the cell. Extracellular fluid is going to lie outside the cell. 2 types of extracellular fluid, ECF, right? Interstitial fluid, that's going to surround most of the cells. And then you have the plasma, which is going to be the extracellular fluid inside the blood vessels, okay? So our body compartments, they're going to be separated by membranes, right? So if we look at the heart, right, this pericardial sac, this pericardial sac is going to be a tissue that surrounds the heart. So if we look at it a little bit more, that tissue membrane, right? So it's going to have that dense connective tissue membrane, right? And when we're looking at that cell membrane right here, right, that cell membrane is going to be made up of that phospholipid bilayer, and that phospholipid bilayer is going to be that cell membrane, which is composed of the phospholipid layers, right? But it's going to be these compartments. These are going to be separated by membranes, right? We're going to have membranes that's going to be able to protect our organs. That's going to be able to protect the outside of our body too, like our cutaneous membrane, right? So when we're looking at biological membranes, so the cell membrane will separate the cell from the environment, from like the external environment. So there's some functions of the membranes, right? Physical isolation, of course, right? The regulation of the exchange with the environment, so allowing certain things to come in and out of the cell, as well as communication between the cell and its environment. And the cell membranes, they also help with structural support, right? Also aid in secretion, right? Where the cell releases substances into that extracellular space. And so membranes, right, they're going to be mostly lipid and protein, right? And so we're going to look at that fluid mosaic molecule. The fluid mosaic model, right, of biological membranes. Okay? And so this just gives you a composition of selected membranes down here, this table, right? So the red blood cell, made up of 49%, its membrane is made up of 49% protein, 43% lipid, and then 8% carbohydrates. That myelin membrane, that myelin sheath around your nerve cells, 18% protein, 79% lipid, right? It's that lipid layer, and it's only 3% carbohydrate. Now, the inner mitochondrial membrane, right? Only none is made up of sugar. Okay, because remember, it's the sugar that we're using to actually break down, right? And that mitochondria is going to use that energy, but it's going to be made up of 24% lipids, and 76% of its membrane is going to be composed of protein. Okay. So this is the fluid mosaic model of biological membranes. So when we're thinking of our biological membranes, we also have to remember that they're going to be in an aqueous solution, right? So you can't see me, right? But I'm kind of moving my arms in my body in like a wavy position. So for example, you see those thingies that have the air going through them, like at a car dealership. Right, it's those little bodies that's going back and forth. That's how like the cell membrane is in an aqueous solution, and it doesn't move back and forth like that now, right? But it does have some fluidity there, okay? But when we're looking at this biological membrane, let's look at it, right? Of course, it's going to be made up of these phospholipids, right? It's going to be a phospholipid bilayer, so one layer of the phospholipids and then another layer of the phospholipids. Also, what's going to be making up, excuse me, this our cell membranes, right? Notice how we have cholesterol that's going to be embedded in there. Okay, cholesterol is going to be embedded in there, helping with the rigidity of our cell membrane. Okay, we're also going to have these peripheral proteins, right? These peripheral proteins, these are going to be on the outside of that cell, or on the surface of that cell membrane. We have these integral proteins, right? They are going to be integrated within that cell membrane. Now, these peripheral proteins, though, they can be removed without disrupting the integrity of the membrane. But like these integral proteins, like this one and this one right here, these integral proteins, right? It's going to be hard, right, to get them out of the membrane. So you're going to disrupt the integrity of that membrane to get this protein out. If we wanted to study this protein, right, and we want to take it out of the membrane to study its function, we're going to have to disrupt that membrane integrity, as well as this transmembrane protein, right? These transmembrane proteins, they span the length of that whole cell membrane. Again, we're gonna have to disrupt the cell membrane to get and to be able to study this. Okay. Now we also have that helps with the rigidity, the fluidity of the cell membrane is the cytoskeleton, right? The cytoskeleton, right? That's going to help with the rigidity, the fluidity, the structure of the cell membrane, of the cell as a whole, really, right? And notice how we also have these sugar molecules on there too, right? Remember that sugar plus the protein is going to give us a glycoprotein, right? And we also have some sugar that's going to be associated directly with the lipid, right? And those are going to be glycolipids, right? Again, these glycolipids, these glycoproteins, they're going to be very, very important for cell signaling, cell communication, things like that, okay? And so lipids, right, making up this cell membrane, we have lipids, right? Cholesterol, phospholipids, sphingolipids, right? So we saw the phospholipids. Sphingolipids are just fatty acid tails. Their heads may be either phospholipid or glycolipid, right? They're a little bit longer too than regular phospholipids too. And cholesterol, right? These are going to be the lipid components of the cell membrane. Okay, you have a carbohydrate component, right? Where that sugar is going to be bound to either a lipid or a protein, right? Forming those glycolipids and those glycoproteins. Again, that's going to function for structural stability, right? Cell recognition and immune response. And we have proteins that's going to make up the cell membrane as well. And they can also be bound to sugars, right? Helping again with cell recognition, so cell communication, structural stability, things like that. Okay. So when we're looking at the intracellular compartments, right, what's inside the cell, right? Each cell in the body is going to inherit identical genetic information in its DNA, but no one cell uses all of this information though, right? So we have differentiation. So there's selected genes that are going to be active, and that's going to lead to specialized cells, right? So cells are going to be divided into compartments, right? So we have the cell membrane that's going to surround the cell, right? Isolating the intracellular components from the extracellular components. We have the nucleus, right, which is going to house our genetic information. Okay, and we have the cytoplasm, right? And within the cytoplasm, we have the cytosol, right? The cytosol is going to be that fluid portion of the cytoplasm. We have these membranous organelles, such as the mitochondria, endoplasmic reticulum, Golgi apparatus, lysosome, peroxisomes, things like that. We also have inclusions. These are going to be non-membranous organelles, so like lipid droplets, glycogen granules, ribosomes. So like we have like energy storage molecules, and we have some like ribosomes. Excuse me. Those are going to be inclusions. And then we have protein fibers that also help to make up the cytoplasm, right? So like the cytoskeleton, right? The centrioles, cilia, flagella, things like that. These are going to be insoluble protein fibers. Okay, so these inclusions that I mentioned, right, like the lipid droplets, the glycogen granules, these are going to be for nutrient storage. They're going to lack a membrane, so they're going to be non-membranous. And then we're also going to have, non-nutrient storage function inclusions such as ribosomes, right? Now, ribosomes, again, they're going to be made from protein, or they're going to be made from RNA and protein, right? But the function of a ribosome, remember, they synthesize proteins. These are the sites where proteins are made, right? Now, we can have fixed ribosomes. Fixed ribosomes are going to be attached to the endoplasmic reticulum. And then we have free ribosomes. These are going to be just within the cytoplasm, not bound to the endoplasmic reticulum. And then we have polyribosomes, right? So many ribosomes on an RNA strand, okay? And so this just goes over the membranous versus the non-membranous organelles that we can see, right? Membranous organelles, again, mitochondria, nucleus, ER, Golgi apparatus, lysosomes, peroxisomes, right? And so definitely remember the function of these as well, okay? We definitely want to remember the function of these organelles, right? Whether they're membranous or non-membranous, okay? So those cytoplasmic fibers, right, they're going to come in three sizes, right? We have microfilaments, which are going to be small. These are going to form the network just inside the cell membrane, okay? Such as like actin fibers, things like that. They're also going to be associated with like myosin for like muscle contraction. We have intermediate filaments, right, such as keratin and neurofilaments, right? And these intermediate filaments, they're also going to be function in the cytoskeleton, also within hair, nails, protective barrier for the skin, right? The keratin. We have microtubules, right, such as the tubulin protein, right? These are going to be very, very small, right? But again, these tubulin microtubules, even though it's micro, right? They're all micro for sure, but these are going to be rather large cytoskeleton fibers. These are going to be the largest of the cytoskeleton fibers, actually, and they're going to help with the movement, right, of cilia, flagella, movement of those chromosomes to their specific places within the cell, as well as intracellular transport of organelles too. So these microtubules, very, very important. Okay, so these microtubules, again, they're going to form centrioles, cilia, flagella, that centrosome, if we remember that centrosome that lies directly like next to, outside of that nucleus. All right, the centrosome is going to be the cell's microtubule organizing center. So we're going to be able to assemble that tubulin monomers into microtubules. Okay, the centrioles, right, which is going to be within that centrosome, is going to be able to direct DNA movement in that cell division. Okay, directing the movement of those chromosomes. We have cilia that helps with the fluid movement across the cell. And we have the flagella or the flagellum, right? Specifically in sperm, right? Helps with the movement through a fluid, right? Helps that cell to move. Okay. Excuse me. Now the cytoskeleton is going to be a changeable scaffold, right? So it can change, and it's going to, again, that cytoskeleton will function in cell shape, internal organization, intracellular transport, assembly of cells into tissues, helping that cell with movement, right? So there are some motor proteins that will create movement as well. So these motor proteins will use ATP to create movement along the cytoskeletal proteins. So you can kind of see like this myosin, this motor protein right here, literally holding this organelle, walking along the cytoskeleton fiber, right? In the presence of ATP, right? So we have these motor proteins, right, that create movement, such as myosin, that helps with muscle contractions, right? The kinesins of the dyneins, right? Movement of vesicles along these microtubules. These dyneins, right? Movement of the cilia and the flagella, right? For their whip-like motions. Right? Organelles, they're going to create compartments for specialized functions. So these organelles, they're kind of like mini organs within the cell. Like how our organs in our body do specific functions, our organelles do specific functions, right? Now, organelles, right, within a compartment, they can have one or more phospholipid membranes, so such as the mitochondria, right? Double membrane structure. Mitochondria has mitochondrial matrix, it has mitochondrial DNA, and it has an intermembrane space as well. Now that mitochondria, remember, a central role in cellular ATP production, right? Cellular respiration. We have the endoplasmic reticulum. Okay, we have the rough ER. Okay, the rough ER, remember, it's going to be able to synthesize proteins. Remember, the rough ER is rough because it has ribosomes affixed to it, right? And again, ribosomes are the sites of protein synthesis. So these rough ER, this rough endoplasmic reticulum, is going to be the site, again, for synthesis of proteins. Now we have the smooth ER, the smooth endoplasmic reticulum, portions of the endoplasmic reticulum that is not affixed with ribosomes. This region of the endoplasmic reticulum is going to be the site of synthesis for fatty acids, right? Steroids and lipids, right? So remember, smooth endoplasmic reticulum, site for lipid synthesis, okay? We also have the Golgi apparatus or the Golgi complex, right? That's going to help to sort, modify, and package proteins into vesicles for them to be transported, right? Whether they're going to be transported so they can be transmembrane proteins, so they need to get within the cell membrane, or they need to get out of the cell, right? They're going to be packaged and sorted via that Golgi apparatus. And then we have vesicles for storage and vesicles for secreting things. So we have lysosomes. Lysosomes will break down bacteria and old organelles. And we have peroxisomes. That's going to break down fatty acids and toxic materials. Right? This just goes over the organelles that I mentioned, right? Mitochondria, Golgi apparatus, endoplasmic reticulum, the peroxisomes, lysosomes, centrioles. Right? Notice how that centriole is within that centrosome, right next to that DNA right next to the nucleus where we house our DNA at, right? And it's those centrioles, right, that are going to be made from microtubules, and they're going to direct the DNA movement during cell division. So they're going to direct those chromosomes to move in their places during cell division. So the nucleus, right, is going to be the cell's control center, right? So within the nucleus, we have it's going to be a double membrane structure, right? We're going to have a double membrane of nuclear envelope. Right, that nuclear envelope is going to contain two membranes with pores, right? And it's going to have these nuclear pores, which is going to be able to, we're going to have communication between the nucleus and the cytoplasm. Within the nucleus, we're going to have chromatin, which is just simply DNA and histone proteins. And we have nucleoli, right? Nucleoli, or the nucleolus, that concentrated portion. So when you look at the nucleus under a microscope, where you see it's really dark in certain portions, it's just concentrated DNA and RNA. And these are the sites where we are making ribosomal RNA. Okay. So this is protein synthesis, right? And we can see how it demonstrates subcellular compartments. So for this right here, I really want you to remember really the central dogma of life. And so that's basically how DNA gets converted into RNA, and that RNA then gets converted into protein, right? And we'll hit on this a lot more for sure. And so DNA is going to be transcribed into RNA, and RNA is then going to be translated into proteins, right? And so this, I'm going to put it in a different color because this is a really important concept, right? This is going to be the central dogma, right, of life, right? This is how DNA directs the synthesis of proteins. So let's zoom in a little bit, right? So we have our DNA, right, being transcribed into making an RNA transcript, right, or an RNA strand. Okay. So number one, after we are able to go through that process, so let me just give you a quick little rundown of it though, right? So DNA gets replicated, right? So we have DNA replication to make copies of itself, right? So now that we have a lot of copies of the DNA, right, and made a copy of itself, now we can transcribe it into RNA. Just another language, basically, okay? So we can transcribe this DNA into RNA. Right, now that we made an RNA strand, right, now that RNA strand will then leave the nucleus, right, via that nucleopore, right? And so the RNA leaves the nucleus and attaches to a cytosolic ribosome, as you can see right here. It can attach to a cytosolic ribosome, right? And that will initiate protein synthesis, okay? And so you can see, if we're looking at this path right here, right, so some proteins are released by free ribosome, so this is going to be a free ribosome, right? So some proteins are going to be released by free ribosomes into the cytosol, or they're going to be targeted to specific organelles, right? So this is going to be targeted to peroxisomes, or it's going to be targeted to a mitochondria, or it's just will lie within the cytosol. Other times, right, the ribosome that are attached to the rough endoplasmic reticulum will direct the proteins destined for packaging into the lumen of that rough ER. So these ribosomes that's on this endoplasmic reticulum, the rough endoplasmic reticulum, will direct that protein to go into the lumen of the endoplasmic reticulum, right? And then it's the proteins that are now, they're going to be modified as they pass through the lumen of the ER, okay? As it passes through the lumen of the ER, we're going to have transport vesicles that's going to move the protein from the ER to the Golgi apparatus, right? And from the Golgi apparatus, we're going to have the Golgi cisternae that's going to migrate toward that cell membrane, right? So the pushing that protein is getting closer and closer, basically, to that cell membrane, okay? And we can see here, right, where some vesicles will bud off the cisternae, and they're going to move in a retrograde or in a backwards fashion, basically, as we can see right here, okay? And then some of the vesicles, they're going to bud off to form lysosomes or storage vesicles, okay? And then other vesicles will become secretory vesicles, and they're going to release their contents outside the cell, right? So those proteins that need to get outside the cell, okay? Or some of the proteins that need to get within the cell membrane, okay? But we'll definitely talk about this a lot more in class, okay? So when we're looking at different tissues of the body, like histology, again, remember histology is going to be the study of tissue structures and functions, right? Cytology is the study of cells. The extracellular matrix is going to have many functions, right? Synthesized and is going to be synthesized and secreted by the cells, right? The extracellular matrix is going to be outside the cell, right? Its composition is going to vary between, tissue to tissue, but we're also going to contain proteoglycans, right, within that extracellular matrix, and these are just simply glycoproteins, and these glycoproteins are proteins covalently bound to a polysaccharide chain, right? So it's a sugar component, polysaccharide, to a protein component, right? They also can contain, this extracellular matrix also contains insoluble protein fibers such as collagen, fibronectin, and laminin, right? And these, again, they're going to provide strength and anchor cells to the matrix. Cell junctions, right? These are going to hold cells together to form tissues, right? So we have these cell adhesion molecules. These are going to be membrane spanning proteins, right? Such as the gap junctions, right? These are communication junctions, right? And we have tight junctions. These are occluding junctions or anchoring junctions, right? So these gap junctions, they're going to permit material to pass from cell to cell. Tight junctions, they're not going to permit things from going from cell to cell, right? So those communication junctions, right, such as gap junctions, right, they're going to allow direct cell to cell communication, right? So basically stuff can go through these pores, right? Little small ions, metabolites can go through these pores, right? These little tunnels. Now these occluding junctions, such as tight junctions, right, they're going to block movement of material between cells, right? So they're going to exclude stuff from moving from cell to cell. And then we have these anchoring junctions, right? And these hold cells to one another and to the cell or the extracellular matrix, such as a desmosome, right? The desmosome is a cell-to-cell anchoring junction, right? Connecting cells with one another, right? Or like a hemidesmosome, connecting that cell to that basement membrane or that extracellular matrix, okay? So we have 4 types of tissue, right? 4 tissue types. We have epithelial, connective, muscle, and neural tissue, okay? So epithelia, right? We have 1 or more layers of the epithelia. They're going to be separated from an underlying tissue by that basal lamina or that basement membrane, right? So epithelia, right? We know that epithelia can provide protection and regulate exchange, right? They're going to be either tight or leaky. And that's going to depend on how easy a substance can pass from one side of the epithelial layer to another side. So leaky epithelia, such as anchoring junctions, that allow molecules to cross the epithelium, bypassing through the gap junction between 2 adjacent epithelial cells. And then we have tight epithelial, and this is where the adjacent cells are going to be bound to each other by tight junctions that create a barrier preventing substances from traveling between adjacent epithelial cells. So nothing's going to pass between these cells, right? You have the structural classification of epithelia, right? So sheets of tissue that cover surfaces or align the hollow organs and tubes. You have secretory epithelia that synthesize and release substances, all right? You have simple, 1 layer, stratified, 2 or more layers. You have squamous, flattened, cuboidal, look like a cube. Columnar, looks like a column, right, of epithelia. You have exchange epithelia, right? Transporting epithelia, ciliated epithelia, protective epithelia, and secretory epithelia. The different types of epithelia tissue, right? Exchange epithelia, very thin, flattened cells, right? They allow gas exchange, right? They're going to line the blood vessels and lungs. These are going to be classified as simple squamous epithelium. And we're going to call the endothelium, it's going to be called endothelium in the heart and then the blood vessels, right? Very, very thin for gas exchange. Transporting epithelium, right? There's going to be cuboidal or columnar. Okay, so we're going to have the apical membrane is going to face the lumen, and that basolateral membrane is going to face the extracellular matrix, right? And this is going to be in the form of cell junctions, right? We're going to have many mitochondria there. But they're going to differ depending on where the epithelium is found, and they can be regulated and modified in response to stimuli, right? But they're going to be able to transport things, right? Can we make that a little bit darker or thicker, right? We're going to be able to transport things through that cell, okay? Then we have ciliated and protective epithelia. Cilia is going to move fluid and particles in the respiratory system and the female reproductive tract, right? We have protective epithelia that prevents exchange, right? And protective is going to protect areas subjected to mechanical and chemical stress. We have secretory epithelia that's going to produce and secrete a substance into the extracellular space. And so it may be scattered or grouped into glands, right? So we have exocrine glands, release their products to the external environment through a duct. And we have endocrine glands that's going to release their hormones into the extracellular compartments, like into the blood supply, right? And then you have the 4 types of tissues, right? So epithelial tissue, connective tissue, muscle tissue, nerve tissue. right? And some unique features of them, like where they're going to be located to. Epithelia cover body surfaces, line cavities, and hollow organs, right? Tubes, secretory glands. Connective tissue support the skin, other organs. Cartilage, bone, blood, all types of connective tissue. We have muscle tissue making up the skeleton, right? Make up skeletal muscles, hollow organs, and tubes, right? We have nerve tissue, right? Obviously, that's going to be throughout the body, concentrated in the brain and the spinal cord, though, right? But we also have those cranial nerves and spinal nerves, right, of the peripheral nervous system. That's going to be throughout the body. When we're looking at the connective tissue, right, connective tissue is going to be made up of cells and matrix, right? Those cells can be either mobile or fixed, right? Such as mobile blood cells, right? Fluid connective tissue, or fixed macrophages in a specific spot, right? Adipocytes, fibroblasts, right? And so you have the different types of connective tissue down here, like loose, dense, adipose, blood, cartilage, bone. They're ground substance, main cell type, and where they're found for sure. Remember muscle and neural tissue. These are going to be excitable type of tissue. Muscle tissue is going to be contractile, right? We have force and movement. So there's going to be 3 types of muscle tissue. Cardiac, smooth, right? Involuntary control. And then we have skeletal, voluntary control, right? And we have neural tissue, right? Neurons are these nerve cells. They're going to be able to send electrical signals, right? These are the excitable cells. And we have the glial cells, right? Outnumbering those neurons, right? Providing that support, right? And remember those neuroglia too, okay? And then, of course, with tissue remodeling, cell death, right? We have necrosis. Necrosis is going to be death by injury, right? And then we have apoptosis. That's going to be programmed cell death, right? So cell suicide. We have stem cells being created through like mitosis, totipotent, pluripotent, multipotent stem cells. They exhibit plasticity, where they can result in differentiate into specific type of cells and specific type of functions they'll have. Organs, right? Organs are going to be groups of tissues with related functions, right? For example, the skin, as an example of an organ, incorporates all 4 types of tissue, right? And they have multiple functions, okay? And just to show you again, right? This is going to be the layers of the skin. We have the epidermis, the 5 layers, or 4, right? Layers of the epidermis. Then we have the dermis, consisting of the 2 layers, right? And then we have under the dermis, we have the hypodermis, right? And so you can see the connection between the epidermis and the dermis, right? Look at these connections right here. Look at this hemidesmosome that's tying that epidermal cell to the fibers of the basal lamina right here. And then, of course, this is going to be the basal lamina right there. So this is just an introduction to Chapter 3, compartmentation, cells, tissues, and organs, okay?